Today I'm excited to look at a biology use case using the clinical knowledge graph built on Neo4j and visualized using the Hume knowledge graph platform. Specifically, I'm interested in how a particular cancerous protein interacts with other proteins, and I want to identify which of those other protein interactions pertain to enzymatic activity. And so with that, let's dive in and see how we can learn more about those identified proteins. I'm going to start by looking up my protein in question, ERBB2. And here we have all the isoforms of ERBB2. Let's bring those together into a single group so we can see them more easily. And now that I have my group of proteins, let's expand its protein-to-protein interactions to create a full PPI network. We'll start with the outbound PPIs first within our database. And now bring in any inbound PPIs. And now we have a full protein-to-protein interaction network. Next, to see which biological processes are linked to these proteins, I'll enable AutoLink and then expand the biological processes for my ERBB2 group. Now we have an interesting set of graphs with our proteins that interact with my biological processes on the left and everything else on the right. So let's clear out the other proteins to refine it just down to those which share my biological processes. Particularly here, I'm interested in enzymatic activity. So I'm going to do a local search for any biological processes pertaining to enzymes. And here I have an enzyme-linked receptor biological process. Let's get all of the proteins that pertain to that biological process. Let's clear out any extraneous related biological processes. And now I'm down to just those that I'm interested in investigating. So let's take a look at these JAK proteins and see how they look. Here I'm reaching out to an external tool that we're able to surface as an iframe within Hume. This allows us to actually investigate the 3D structure of a given protein and evaluate other auxiliary metrics that might be relevant to a researcher. We can do the same for JAK2 and examine its 3D protein structure and take note of any other additional information that this tool might offer us. Next, I want to look at the drugs that have been associated with these. So I'm going to look at my Show Target Drugs action. Let's pull up what drugs have been associated with these genes. And now I have a list, and we can see there's some overlap between the two genes. Let's go back and rerun my action using my intersection filter so we can get just the drugs which interact with both proteins. And now I have my diagram of drugs and proteins. So we can do a couple things with this. So first, I'm going to select all of my proteins and generate a report that I can then export and potentially share with other people. So here are all of my proteins, genes, and drugs. We can export this as a CSV or an Excel file. I can also add my individual proteins that I'm interested in to a collection. So here I'm going to add them to my targets to test collection, where I might keep any potential protein targets that I'm interested in. And lastly, we'll use Hume's snapshot feature to capture this visualization. So I'll call this my enzyme exploration and give it a description, and then click Save. And now I have this available to view as an individual snapshot of the data I've found. So here I can go in and move things around and have a static image of the visualization I want, or create a shareable link and then drop this in a Slack channel or Teams or an email and share it with other researchers who might be interested in the finding that I've discovered. So here we started with a particularly interesting target protein. We looked at its vast PPI network and then narrowed that large number of proteins down to just a few that meet the criteria that I'm interested in investigating. So I hope this might spark some ideas for how you might apply Hume and Neo4j into your drug discovery workflows or scientific research, and we'll be really interested to hear what you do.